Our next speaker is Dr. Larry Beck. Dr. Larry Beck is from Boston Medical Center, where he completed his nephrology training. And as I mentioned earlier, he was first author on the 2009 New England Journal of Medicine paper that really took this field by storm. And he'll talk about the discovery of that. And also want to point out, I looked to see, I was curious, how many citations has that paper had? And on Google Scholar, it's already up to 1,800 citations in about 10 years. So really impressive. You want to share your screen?
Okay, sorry. I'm going to be the first of 3 Tiffany Kazas, I think. This is why you're not supposed to share your individual links, Tiffany.
It's okay. We got you renamed. All good.
Oh,
yeah.
Perfect.
We had about 50 Samira Farouks on the day one.
Nice.
So.
Okay, well.
I'll give a 5-minute warning and a 1-minute warning.
Okay.
Live.
Good. So yeah, I put the discovery and use of PLA2R and THS7DA in 30 minutes or less because it's tough to talk about this. I usually get 40 to 50 minutes, but thank you for inviting me. And I was very excited by Dr. Arthur's introduction this morning. And I would really like to be invited to the 2024 meeting because I've never seen a solar eclipse. And I guess the dance we did this morning will put the path through Little Rock again. So I'm happy to, hopefully we'll have some more new antigens by then. These are my disclosures. And I'm going to start the talk with, talking about the antigen discovery process and then how we established the paradigm, or we and many others around the world established the paradigm with PLA2R antibodies. Then I'll move on to THS7DA as a less common antigen antibody system with similar principles. And then the subsequent talks by Dr. Al-Rabadi and Kaza will talk about some of the newer antigens, and then I'll wrap up. So we're fortunate enough to get kidneys that can't be transplanted and are given to us for research purposes from the New England Donor Services. And what we do is we take kidney cortex, we mechanically sieve the glomeruli, and then we can make detergent extracts and run those by gel electrophoresis. And then we had back in the early 2000s, we had a big repository of patient serum, idiopathic membranous nephropathy, and we used that serum to blot for glomerular proteins. And we found this common band that wasn't found in FSGS, diabetics, healthy controls, other autoimmune diseases. And this is what eventually became PLA2R. Other people had done this since the 80s when we started looking for the human antigen. Some of the reason that we were successful, I think, many Western blots are run with reducing agents, and the antigen PLA2R, the antibodies don't detect PLA2R if it's reduced. So we did non-reducing conditions, and IgG4 is the predominant subclass of antibodies found in the kidney in idiopathic or primary disease. So we limited our detection to only human IgG4, and this gave us a very nice clean band that allowed us to see that there was something there. Now, seeing a band doesn't mean you can easily tell what it is. So we tried to partially purify the protein. It was a glycoprotein. If we took off the sugars with this glycosidase, it shifted down in size. We tried to partially purify it on lectin columns. And ultimately, we cut out the protein backbone in these gel regions. We sent them to our colleagues at Louisville. We didn't know John Arthur yet, who did the mass spec for us. And we evaluated the list of proteins that came back. And there were lots of interesting proteins in that size range. We had angiotensin converting enzyme, podocolixin, nephrin, alpha-3 integrin, all of which are important kidney proteins. But when we got antibodies to these specific proteins, we saw that they weren't quite the same size in our glomerular extracts. So we kept hunting. So this is the untold story. And if you want to read more of this, there's a paper by Dr. Truong in referenced here that goes through a little bit of the story. What actually happened? We never would have found PLA2R on the list because it was way, way down. So I kept looking for ways to get PLA2R to a different size, and thought maybe it's proteolytically cleaved from the surface of the podocyte. So we got as fresh glomeruli, human glomeruli as we could. We put them into a saline or a tissue culture solution. We just sort of heated them at 37 degrees to allow any shedding to occur. And we spun down the glomeruli. We collected the supernatant to see if anything had been released, maybe a cleaved piece of PLA2R, but there was still the full length protein here. So we thought, okay, maybe we need to ultracentrifuge this at 100,000 times G. So we did that. Interestingly, we did get a tiny little membrane pellet, but the antigen was still in the supernatant, probably exosomes or something that hadn't fully spun down. But when we took that high-speed supernatant and ran it on the gel and then cut out the band, the most abundant protein spectra were from PLA2R. So that's when we got in touch with our colleague, Gerard Lambeau, who we didn't know at the time, but we know that he had cloned this protein called the M-type phospholipase A2 receptor. I emailed him, I told him what we were up to. He was very gracious and sent us a huge package on dry ice across the sea with antibodies, recombinant proteins, and pretty quickly we realized that this PLA2R was in fact the antigen. And this is an early schematic of the M type. You may see that in print. It's the muscle type phospholipase A2 receptor because it was cloned from rabbit muscle. This is a northern blot, these things that we used to do to look for RNA 30 years ago. And you can see the rabbit muscle has a lot of the PLA2R message. Interestingly, in humans, the predominant tissue that has this mRNA is the kidney. And this is pretty much the glomeruli. So it sort of fits with what we were finding. This is in our paper, we had a extended view of the protein. It's actually a very convoluted double ring structure, pH dependent, but there's this N-terminal cysteine-rich domain. That's where the immunodominant epitope lives. Everyone who has antibodies circulating PLA2R targets this cysteine-rich domain. Then there's a fibronectin-2 type domain, and then 8 C-type lectin-like domains. And we know that there are other epitopes in CTLD1, 7, and 8. And then, like nephrin, as Dr. Wines talked about, there's a transmembrane region and a short cytoplasmic tail. So some of the early observations we made in terms of antibody presence in the blood and clinical activity were that it was, we'll start on this side. So these were some samples we got from Jack Wetzel's labs, and we were blinded to the timing of them. But when we broke the code, we could see that patients who were nephrotic had high levels of anti-PLA2R. When they ran into remission, their antibody levels dropped to 0, except for this one case, which we don't really have a good explanation for. And then when these same patients relapsed, their antibody levels came back up again. Another patient who was representative, this was anti-PLA2R detectable in 2005, but as the patient went into immunologic remission, that was followed by a clinical remission. So the antibodies are gone, and then the protein is coming down later, the albumin's going up. So these were our early findings. There was another very nice paper by Pierre Ronco and Hanna Debiek as a letter to New England Journal that showed not only should we be looking at the serologies, but we should also be staining the tissue in the patients, because in many patients, there will be a seropositive for anti-PLA2R, and their tissue will stain positively in the deposits. There are some cases that don't stain. This may be due to masking of some sort. There are clearly some cases that, and we'll hear about these later in the second half of this session, that are PLA2R negative, both in the tissue and the serum. And then there are these cases that, even though they didn't have antibodies in the serum, they were tissue positive for PLA2R. And I think many of us regard this as a sort of a historical remnant of a prior immunological episode. And why do we think that? Well, there's this nice paper from China from 2017, and they only looked at patients who had tissue positivity for PLA2R. There are about 500. And then 3/4 of them had anti-PLA2R in the serum, but then 25% didn't, like this, this last case. So tissue positive, serum negative. And it turns out that this group, the dotted line, these are the ones, they start with lower proteinuria, they start with a higher albumin, and they're the ones that reach clinical remission much more quickly than the group that's seropositive and tissue positive. These guys still have a long way to go. Excuse my cough. And then there's a nice paper by Chris Larson from Little Rock and Arcana Labs, nephropath at the time, I think, who showed that the staining for PLA2R in a membranous biopsy was pretty specific for PLA2R. So 75% of idiopathic cases had PLA2R staining, but there were some secondary cases, not in lupus, not class 5 lupus nephritis, but for example, sarcoidosis, cases of cancer-associated hep C that did stain positively for PLA2R. And the question is, and we still don't know the answer, is something about sarcoidosis or hepatitis inducing an autoimmune attack, or is it just coincidental disease? One of the reasons we think there may be coincidental diseases, this was a study done by Weisong Qing from Nanjing University, who came to our lab with a lot of Chinese samples. He also showed that 80% of idiopathic cases were seropositive for PLA2R. And most cases for lupus or hepatitis B or cancer-associated disease were seronegative, but there were some seropositive cases. When he went back and looked at and restained the biopsies for IgG4, it turns out that these particular cases were IgG4 predominant, suggesting that these may be a primary disease and the patient had coexisting ANA positivity or evidence of hepatitis. So we think there can be coincidental cases and not necessarily that these are secondary causes of membranous. Shane Bobart and the Mayo Clinic crew have suggested that maybe we don't even need a biopsy and came up with an algorithm. This is Jürgen Flürgi's report in Kidney International and the Controversies Conference for KDIGO, that if you have a nephrotic patient who has seropositivity for PLA2R, they're low risk, meaning their GFR is good, there's no evidence of secondary features, then you can probably make the diagnosis without a kidney biopsy. If they're higher risk, if they have potential secondary features, if you want to start high risk immunosuppressive therapy, then probably we should biopsy that patient. So the clinical use of serology to monitor and predict disease course is really based on a key principle, that there are two disease courses, the immunological or serological course as monitored by circulating anti-PLA2R, or in other cases, say anti-THS7DA, and then the clinical course, which is what we see and what we had to rely on 15 years ago by proteinuria, edema, et cetera. And this was a graph we came up with more than 10 years ago, and shows that there's this immunologic phase, the anti-PLA2R comes down and disappears. This was what we would call immunological or serological remission. And then followed sort of with a lag time, the proteinuria will come down and some people will be left with a partial, some people will go on to a full complete remission. This graph came because we had done these studies that were pilot studies that Fernando Fervenza and Dan Catron had done, treating patients with idiopathic membranous nephropathy with rituximab, and had saved a lot of those biosamples. So for example, this is one patient whose blood at time 0, 3 months, 6 months, 9 months, et cetera, we tested by Western blot for anti-PLA2R. You can see that if we graph it from 100%, the anti-PLA2R comes down and is gone by 12 months, which is probably around here. But the proteinuria in this patient came down, with a lag time of 6 to 9 months. And if you looked at everyone in aggregate, we saw the same phenomenon. The anti-PLA2R came down and disappeared by an average of 9 months, but the proteinuria was coming down, but it was still nephrotic at 9 months and continued to decline. So it's not just rituximab. There have been lots of studies since then that no matter what whether this is a treatment-induced remission with the Ponticelli regimen or with cyclosporin or rituximab or a spontaneous remission, there's this similar lag time between the antibodies and the decline of proteinuria. So why is this the case? And we had thought about this. I mean, we had our ideas, but we finally got this submitted, and this was hard work by Gabriel Lerner, who's in Little Rock and is on this call now. So congratulations, Gabriel, getting this thing out. But we came up with a conceptual way to think about the antibodies and the formation and dissolution of the deposits and why that would lead to clinical lag time. So I urge you, all of you who have access to Kidney International, to go look for this and read it. This was one of the figures from the paper, and we know that the Ehrenreich-Schurg staging of stage 1, stage 2, 3, 4. 4 is the deposits that have become electron lucent and are starting to dissolve essentially. They actually start as a even smaller, Rodriguez from Mayo Clinic has called this a stage 0 deposit, and really we can only find these in the transplant. And this was a case that we had at our center where the pathology showed, because immunofluorescence is more sensitive, that we saw the deposits by immunofluorescence, but we didn't really think we saw anything by EM. But then Dr. Henderson, our pathologist, came back and said, I think there are these tiny little deposits under the slit diaphragms. And over time, those went on to form the stage 1 and stage 2 deposits. This is a different patient, but years later, they might look like this. It was interesting because this is our patient with the deposits below the slit diaphragms. This is a Hayman nephritis rat from 1978 that shows the same localization of anti-megalin antibodies in this experimental model. So probably the first sign of deposits can be right under the foot process, right under these slit diaphragms, maybe with this extra fluid flux that you saw in Dr. Wine's talk, that it's going through these areas. We came up with these phases of disease. Initially, the immunologic initiation, we don't know what starts this process, the loss of tolerance, but for some reason, B cells start to make autoantibodies. At these very low levels, they're probably all caught in the kidney. So this is the kidney as a sink hypothesis, where there is immunologic activity, but you might not detect the circulating antibodies because they're all binding to PLA2R in the kidney. Eventually, though, those deposits start getting bigger and bigger, and they become saturated. So you start to see some autoantibody in the circulation. And then over a lag time, you get, because it takes time for these deposits to build up to cause the complement-mediated damage to the podocyte. You get proteinuria coming up with a lag. There's an active phase of disease where you have everything there, antibodies, deposits, proteinuria. Then either spontaneously or with treatment, you start treating the immunologic condition. Antibodies go down and disappear. This is the immunologic recovery and then immunologic remission, during which there's clinical recovery and finally clinical remission, whether that's a partial remission, less than 3.5 grams per day, or a complete remission less than 0.3. And others have come up with serological based algorithms. This is Anne DeVries's and the Mayo group and De Classic, who looked at, if you have a patient with a certain titer, are they nephrotic? Is the titer going up? To decide whether or not you want to start treatment. If they're not nephrotic, the titer's going down, you might just want to wait and see if they go into a clinical remission. We're still struggling. We don't know what the final KDIGO GN guidelines are. I think they're going to look something like this in terms of treatment. The Dan Catron's Toronto algorithm, sort of less than 4 grams, 4 to 8 grams is moderate risk, high risk is greater than 8, have been the PLA2R has been added to that. I don't necessarily agree with a threshold of 50. I don't think there's a great threshold we could define, but moderate risk, if you can't get the protein down under 4 grams, if they have significant anti-PLA2R that's not going down, these are probably moderate risk and should be treated with an agent like rituximab, calcineurin inhibitors. The higher risk patients may have very high levels. They may be losing kidney function. Rituximab can work, but you might want to consider an alkylating agent, corticosteroid therapy, which does work faster. Whoops. We know that about 25 or 30% of cases will relapse. We see the same sort of, the patient can be in immunological remission and then be in a clinical remission, and then the antibodies come back, so they have an immunologic relapse followed by a proteinuric relapse. So the same thing happens, and we've seen this in our patients, and many of you have probably seen this too. I'm not going to talk about recurrent membranous nephropathy, but the same thing happens. A patient can come into the transplant period with circulating antibodies, a fresh kidney is put in, and that fresh podocytes are being sort of now targeted by these anti-PLA2R antibodies. Often the transplant immunosuppression is enough to get rid of them, but if you come into transplant with high enough levels, you can have sort of a full clinical recurrence. And of course, it can come back years later and cause a recurrence, even if you don't have the full recurrence at time of transplant. Okay, so let's see how much time we have. We're going to talk a little bit about THS7DA, which is seen in about 3% of patients. I spent a long time looking at the amino acid sequence and came up with this repeating structure. And with help from Shana Stoddard from Rhodes College, we came up with this structural model of THS7DA with this positively charged region, which may stick to the heparan sulfate proteoglycans in the matrix. Like PLA2R, it's an IgG4 predominant. So many of the antibodies from our patients are IgG4 anti-THS7DA. If you co-localize the antigen THS7DA with IgG4, you get a perfect co-localization. This is a nice immunohistochemistry study from Chris Larson's group. Interestingly, this protein sits right adjacent to the GBM. That seems to be its primary location in the podocyte. And Toby Huber's group from, he's currently in Hamburg, Germany now as chief, but they showed that by immunogold, the THS7DA localized right at the sort of slit diaphragm area. Interestingly, Catherine Meyer-Schessinger group had gone on, she's also in Hamburg, had done these high resolution studies that Dr. Wines had done and showed that, so red is nephrin, this meandering slit diaphragm appearance of nephrin, and the green is THS7DA. So they showed that THS7DA is sitting directly beneath nephrin, which is, I think, fascinating. We don't know why that is or what it does, but I think it's something we'll learn a lot more about in the future. We can use anti-THS7DA similar to anti-PLA2R. Patients who have active disease are often having high levels. Those in complete remission have lower levels. If you follow a patient, the patients that have anti-THS7DA that start high and go low, go from active disease to remission. Those patients that have high levels of anti-THS7DA that don't go down significantly, those are the ones that remain in active disease. That's either with sort of spontaneous remission or treatment-induced remission. I'm not going to talk a lot about epitope sites in either molecule. There's a suggestion that more specificity is targeted by anti-PLA2R means worse disease. We also know that in THS7DA, there are multiple epitopes. We've predicted them structurally in our model, but the Humber group has also made recombinant pieces of THS7DA and shown that the dominant epitope is also N-terminal, but there's a wide scattering of other anti-THS7DA antibodies.
Right, to wrap up, we have about 3 minutes or so.
Yep, okay. I won't go through this slide, but there's a link to malignancy with THS7DA, and there's evidence that the tumor may express it and be the first site of immunization. So we know that there's a link of membranous nephropathy to malignancy, so always think about this. We may hear about this more this afternoon, but certainly THS7DA has been associated with cases of malignancy-induced membranous. And we'll hear a little bit more about NEL1. I just want to talk a little bit about technology. We use this old school method of cutting out bands and sending for mass spec. Sanjeev Sethi from Mayo has pioneered and popularized this method of laser capture microdissection and looking for antigens. And I just want to make the point that advances we make really require, innovative thought and application of new or old tools in thoughtful ways. And I think some of the things that have helped in antigen identification are, we know that membranous nephropathy, at least the primary form, is IgG4 predominant. So that's why we restricted our Western blots to IgG4. We now know that these deposits have built up over months to years, becoming enriched in the deposits. And that's really what Sethi in the Mayo group has shown that has allowed this mass spec discovery process for new antigens. We also know that these antigens are probably complex to IgG in the deposits. And Tiffany Kaza with the Arkansas group has shown that you can actually pull out the IgG with the antigen prior to mass spec. And that's also a very good way to identify antigens. So summary, we know this is the major and a minor antigen in membranous nephropathy. We know the antibodies have a consistent relationship to clinical course, and especially in membranous, which has a very long duration. It's probably worthwhile looking, at immunologic changes as a surrogate for what the clinical outcome or changes are going to be. Finally, and this has been my career, the more we learn, the more questions come up. And even though we've made some very major advances in membranous, there's always more work to be done. So for you, those of you who are creative and innovative and want to go into science, think about the underlying biology. Don't be afraid to be creative and go off the beaten path, because really the future of nephrology is in your hands.